We spend a third of our lives asleep. If our prehistoric ancestors could have survived without sleep, evolution probably would have taken care of this massive inefficiency for us. Clearly, there's something biologically important about sleep, since every creature in the animal kingdom does it. But we don't have to look that far to know how critical sleep is, because, especially here at Caltech, we all know what happens when we don't get enough of it. Yet, even though we spend so much of our time in this bizarre state of unconsciousness, much of the why and how of sleep remain a mystery. The first scientists to begin to tackle these questions have been biologists, who study the genes in our DNA and the circuits in our brain that are involved in sleep. But the genes in our genome and our brain architecture don't change on a 24-hour cycle. What is constantly fluctuating and more directly influences the changes in behavior and brain activity we see during sleep is the molecular composition of our cells. And that's where I come in. As a chemical engineer, I can help paint a more complete and dynamic picture of sleep by zooming in further and helping us understand sleep at the molecular level. The molecules I'm most interested in studying are proteins. Proteins are the molecules in our cells that do things. They're the signaling molecules, enzymes, transporters, structural components, and antibodies. For my PhD, I investigate what proteins cells produce during the night and how that differs from the protein cells produce during the day. To do this, I use the model organism called zebrafish because, believe it or not, we actually have a lot in common with these creatures. Zebrafish have a lot of the same genes known to play a role in sleep in humans, like melatonin, serotonin, and hypocretin. And as early as 4 days old, the larvae start exhibiting patterns of sleep at night and wake during the day, just like us. But when I came to this project, there was no way of picking apart what was being produced when. While we could get a snapshot at a moment in time of all the proteins in the fish, there was no way of distinguishing between which proteins were made at night and which were made during the day. To do this, I use a cool new method from my lab. It works by using a tagged version of the building blocks that make up proteins, which I give to the fish during the time window I'm interested in studying. The fish then incorporate this tag into proteins as they're being made, and the tag then allows me to pull out the newly made proteins from the pool of pre-existing proteins. I can then identify these proteins using an instrument called a mass spectrometer, which allows me to detect exactly which proteins were present in each sample and their relative amounts. Using this technique, I've identified thousands of proteins synthesized during day and night. And this data set, which I've only just started to dig into, will allow us for the first time to identify which molecules are uniquely synthesized during sleep and which during wake. What excites me most, though, about this data set are the questions that arise from it. Which proteins, what is the function of the proteins made at night, and how does that differ from those made during the day? And do proteins made at night directly facilitate sleep, or do they require sleep to be produced? I hope to answer some of these questions, and in doing so, shed some light on the sleep we get at night. Thank you.